Let's welcome Alex Leow as our speaker today. Alex did her PhD and MD, both here at UCLA, and her mentors were Kenneth Lang and Henry Wang in MING, and Ken Lang was in biomathematics. So her research focused on high-dimensional image registration with applications to both structural volumetric data and high angular resolution diffusion imaging HARDI from DWI. Alex now is a full professor at the University of Illinois, Chicago, and also is an attending physician, psychiatrist at the University of Illinois Hospital. So at UIC, she co-leads with Olu Agerelated, the Center for Computational Neuroimaging and Connected Technologies. And this lab is focused on understanding psychiatric illnesses and brain diseases with a variety of diverse approaches coming from brain imaging, non-invasive brain stimulation, virtual reality type, immersive display visualization environments, as well as mobile technologies. So along the way, Alex also co-founded a company devoted to brain health. So Alex most recently leads a BiAffect project, and that project is focused on brain health fitness trackers by way of smartphones. And there's a whole new talk that she'll present tomorrow here at 11 a.m. So please make sure to attend either in person or in Zoom. Alex, I should also add, is a pianist. She plays classical piano and has done solo performances as well as a chamber musician. So today her topic is going to be understanding the excitation-inhibition balance in AD pathology and using some neuroimaging findings. So let's welcome Alex.
We should get rid of the thing on top.
It's not showing up on the computer.
Okay, so you go all the way to the right, click there, and then disable, hide floating media.
Which one is it? I can see from here.
Okay, I can see that. I can see that. No, sorry.
It's also a shortcut.
This one?
No, down.
No, wow. Okay.
All right. So thank you so much for having me today here. And with all the excitement from technical issues we have, hopefully everything will be fine after this. Can everyone hear me? Good. So I am Alex, and my talk today title is Understanding Excitation-Inhibition Balance in Alzheimer's Disease Pathology. And it is going to be a neuroimaging perspective. I did my PhD in biomathematics. I finished in 2003, and then I finished my residency training in 2009. And then after that, I left UCLA. So today is really coming home for me. So it's really special to be coming home. So with that, I want to thank Dr. Joshi for inviting me to do this talk. There will be a second presentation tomorrow, and I will be talking about something that's entirely different. It's going to be about turning smartphones into brain fitness trackers. So I think of my back-to-back presentation today and tomorrow as sharing with you some self-reflection. When I was growing up, I wanted to be a pure mathematician and a concert pianist. And somehow I ended up being a psychiatrist and an applied mathematician. So looking back at my career, how I managed to connect the dots and find a way to connect mathematics, neuroimaging, smartphones and playing the piano at the same time, I hope to share with you this journey today and tomorrow. So with that, these are my disclosures. Oh, should I wear the mask or should I take it off?
I think you can take it out.
Yeah, okay. All right, so hopefully now people can hear me better. And these are the funding sources about what I'm going to talk about today. So I first came across this idea of the critical brain hypothesis or criticality. And for people who are not familiar with the critical brain hypothesis, it is definitely not the kind of critical that you are probably thinking about. There is even a critical brain webpage on Wikipedia. So the idea goes like this. Brain dynamics has to be continuously and dynamically transitioning between two phases. One in which activity will rapidly die, reduce and die, and the other where activity will build up and amplify over time. So right away, we see this dynamic balance between excitation and inhibition. On one hand, if we have too much excitation, activity will build up and amplify over time, and that's of course overexcitation. And on the opposite end of the spectrum, activity will rapidly reduce and die, and that's under excitation. It turns out that this excitation-inhibition balance, or EI balance, is also important in normal brain development. In early adolescence, there needs to be a lot more excitation than inhibition so that new skills can be learned. I borrowed this illustration from this 2014 review article. You can see on the top left, the first arrow is indicating the degree of excitation, and the x-axis is age. And the second arrow here, the dark green, is the degree of inhibition. So you can see that early on, there is, relatively speaking, more excitation than inhibition, so that new skills can be learned. If you look at the graph on the bottom, let's go back. Very similar ideas. When there is more excitation, then it's more about reward seeking, and that's the red line at the bottom. On the other hand, when there is more inhibition, as we get older, there's relatively speaking more inhibition than excitation. Then it's more about executive control and affect regulation. So abnormal excitation-inhibition balance has been conjectured in quite a few neuropsychiatric conditions, including autism, ADHD, depression, and epilepsy is, of course, really a quintessential example of hyperexcitation. But most relevant to my presentation today, there has been recent evidence supporting abnormal EI balance in neurodegeneration, in particular Alzheimer's disease. On a 30,000-foot view, in animal models of Alzheimer's disease, beta amyloid induces synaptic hyperexcitation by suppressing inhibitory transmission. And that synaptic dysfunction may represent the earliest changes in Alzheimer's disease. And also, I wanted to make sure I have enough time to cover how many, is it 45 minutes?
Yeah, 45 minutes.
Okay, so that means I may have to speak faster. And later on, I'm also gonna connect this observation of hyperexcitation, connect it back to this recognized sex differences in Alzheimer's disease. It is known that women are at a greater risk of developing Alzheimer's disease, and researchers don't really know why that's the case. In the past, people believed that it's simply because age is the single most important risk factor for Alzheimer's disease. The older we get, the more likely we can get Alzheimer's disease. And since women tend to live longer, then maybe that's the reason why women are at a higher risk. But more recently, we believe that the picture is a lot more complex than that. And hopefully we will come back and talk about this later. We do have quite a few slides to cover today. So at some point, I may have to start skipping. The next few slides, I am gonna briefly review the cellular architecture of hippocampus, or the hippocampal network. As we all know, hippocampus receives information and projects information back to the entorhinal cortex, and that's ERC. The major output region of hippocampus is, of course, CA1, and that's at the bottom. CA1 receives information from the so-called indirect and direct pathways. The indirect pathway is trisynaptic in nature. So there are three synapses. The first synapse is, of course, from the second layer of entorhinal cortex to the dentate, and that's the first top blue arrow. The second synapse goes from dentate to CA3, and that's the mossy fiber. Then we have the third synapse that goes from CA3 to CA1, and that's the Schaefer's collateral. What's unique about CA3 is that it has this autoassociative fibers, and these are essentially positive feedback loop. And this will come back later to really understand why hyperexcitation is relevant in Alzheimer's disease. And of course, CA1 also receives direct innervation from a layer 3 of entorhinal cortex, and that's the fourth arrow. And by the way, these slides are given to me by Dr. Arnold Baker at Johns Hopkins, and we'll talk about his amazing research later. He's a good friend of mine. What happened in normal aging is that there is age-related loss in the perforant pathway, and that's the first synapse we talked about that goes from entorhinal cortex to a dentate in CA3. So the dashed lines, the dashed red lines are the perforant pathway, and these are the ones that are affected in normal aging. So this has been shown in animal studies as well as human studies. For example, in this first paper from 2004, you can see there's a gap between the young mice. I believe these are rodents, mice or rats. You can see that the long-term potentiation maintenance, and that's the y-axis, between the young rodents and the older rodents, there is a difference, there's a gap. And this is again in the perforant pathway. This has also been shown in human studies, and this is imaging using diffusion imaging data. Again, what you see is that the integrity in these pathways are somehow degrading in humans as well as we age. And this degree of degradation is actually associated with the performance of a verbal learning task. And that's the line on the right, highlighted using a blue arrow. So that was normal aging. How about abnormal aging? I borrowed this very nice diagram from this 2019 review article, and proposed this working model for ApoE4 induced GABAergic interneuron deficit. This is a working model for how ApoE4 interacts with Alzheimer's disease. As we know, ApoE4 is the most important, most relevant genetic risk factor for late onset Alzheimer's disease. So the idea goes like this. Since ApoE4 changes the way A beta is aggregated and cleared from the system. So with ApoE4, you now have more A beta aggregation, in particular in hippocampus. And then you now have tau pathology. And these metabolites are, of course, neurotoxic. And they are in particular neurotoxic for the GABAergic inhibitory interneuron in hippocampus. So of course, these inhibitory neurons die, then that's going to lead to a network dysfunction in the form of hyperexcitability. And that's going to have downstream deficits in learning and memory. And this is a reason why some researchers, including myself, believe that early on, hyperexcitability is a sign of Alzheimer's disease pathology. And Dr. Baker at Johns Hopkins actually is running a drug trial. And he showed that by reducing hyperexcitability, they were able to improve memory in people with MCI or mild cognitive impairment. And the medication that they are repurposing is, of course, an anti-seizure medication, is Keppra. And they are now running a much larger trial. I just talked to him before my presentation since I wanted to know any more recent update. And as far as I know, the larger trial is, again, statistically significant. There's an improvement if you target hyperexcitability in the hippocampus. So it's very exciting research.
What about the doses? Compared to what epileptic patients get, that's a much smaller dose. How do you try to do about that?
I am not sure. I think they may have done this in a mouse model first, and maybe they extrapolate a dosage from that. But that's again an excellent question. And as far as I know, Depakote has been tried at least in mouse models of excitability. I don't know if it has been tried in human studies yet. But ever since I came across this data, I was fascinated, and everything seems to come together in a coherent story. So that goes to the first part of my talk, talking about findings from my own lab. And hopefully I will have time to talk about the second part that is more computational. The first thing we did was we tried to understand EI balance in the mouse model of Alzheimer's disease. And this is spearheaded by a graduate student who is graduating very soon, Zach Morrissey. He used a transgenic, APP knock-in mouse model that contains 3 different mutations in the gene encoding the APP protein or amyloid precursor protein. For this specific strain of APP knock-in mouse, you see A beta amyloid doses in the absence of APP overexpression. So you don't get a confound from that. And the production of A beta plaques starts at around 4 months of age, but you don't really see any deficits, just very mild behavioral phenotype. You don't see any really deficits in memory or behavior until about 1 year of age. So what has been reported in the literature using this same model of Alzheimer's disease mice is that in this 2019 paper, they showed subtle behavioral changes and increased prefrontal hippocampal network synchrony in this mouse model before any prominent plaque deposition. And this first arrow on the left really shows where the hypersynchrony is. The top half is this specific mouse model, and the lower half is the control mouse model. So you can see that the area that I highlighted that is more red, more synchrony than the control mice. And the second graph plot on the bottom right The y-axis is functional connectivity, and the x-axis is age. Let me see if I can get the mouse to work. Okay, can we see the mouse here? So we have the young, the middle age, and the later age. And the red line is the APP knock-in mice, and the blue line is the control. So you can see early on, there is increased functional connectivity. And then that increased functional connectivity seems to drop towards the older age. So the way I explain this finding is essentially early on, we are overclocking the brain. These mice, their brains working so much harder because of that hyperexcitation. And that eventually leads to the brain burning out at the end. So it's overclocking followed by burning out. And we're going to come back and see the same pattern again later. I'm going to skip this, this other paper using a similar strain. It's a very similar story. You see hypersynchronous bold activity. So this is what he did. He only studied female mice, again, because well-recognized sex differences. We wanted to focus on female mice. And we have also early, middle, and late. And these are the numbers of mice in each group. It's on the top right in the red box. And then we did the standard imaging. It's a 9.4 Tesla animal. We have the structural T2, we have the resting state, and we have the diffusion. And the bottom right is sort of a standard pipeline for co-registering the images to a template and atlas. And these are the sequences. In the interest of time, I am going to pretty much skip. The only thing I want to highlight is that, as you can see, with animal imaging, even with only 2 slices of resting state, the acquisition time is about 40 minutes. And a good percentage of the mice actually died after or in the middle of the scan. So the first thing we can do is on the top, on the far right, we can take all the voxels that are flagged as hippocampus, and you can simply do a voxel-wise Pearson correlation, and that's going to give you a hippocampus functional connectome. So that's the first thing we did. And then we can separate that into intrahemispheric or interhemispheric, and that's the matrices on the right. And the quadrant that is highlighted using the blue arrows, those are the interhemispheric hippocampal connection. The left column is early in the middle, and the far right is the late, the old mice. The top row is wild type or control, and the bottom row is APP knocking. Again, if you look at the same plot that I showed early on, you, I flipped this. Did I flip this? No, yeah, this is good. If you look at the yellow is the APP knock-in. We again see the similar idea of overclocking. In the early age, there is more connectivity, better, higher functional connectivity for the APP knock-in, and that seems to be leading to the brain pooping out later. So we can see the blue line is the wild type or control, and the yellow line is the APP knock-in. Of course, what we can do is we can also do more of a regional analysis. If you look at the matrix representation that I had on the right with the red box, now not only we separate by hemisphere, we also separate by difference of regions of hippocampus. We have CA1, CA3, and dentate. CA2, of course, is very small, especially in mouse models. So usually it's very hard to get CA2. And then we ran some analyses. If you look at the smiley face at the bottom right, you can see again that a crossing I talked about earlier. In the earlier age, the APP knock-in mice has higher connectivity. And then as the mice get older, then the brain seems to be pooping out, and now the functional connectivity is dropping as you go into, after age, 12 months of age. If we look at the specific region and see which one has the largest, this is one thing I forgot to mention. If you look at all the slopes on the bottom left, all the slopes are positive, at least in our study. So regardless of the whether it's wild type or APP knock-in, overall there's a increase in interhemispheric hippocampal connectivity with age. And if you look at the specific region that seems to exhibit the largest connectivity increase with age, and that seems to be CA3, left CA3 to right CA3, and that's the red arrow highlighted here. Let me see if I can bring my mouse. These. And this one, you can see the slope for that on the bottom left. The slope for that is really the highest. And the reason why we see CA3 is coming back to the autoassociative fibers that I talked about earlier. Imagine if the ApoE4 or A-beta plaques are neurotoxic and they really kill the interneuron, the inhibitory interneuron, then where you're going to see the most hyperexcitation, you're going to see in the CA3, because now the positive autofeedback is unchecked, is unbalanced, because the inhibitory tone has been dropped, basically. And then, although we saw interhemispheric hippocampal increase in connectivity with age, we actually see the complete opposite if you look within intrahemisphere. If you look at all the slopes on the left, the red box, all the slopes are negative. And all those are the functional connectivity within each hemisphere for the hippocampus. Furthermore, in the wild type mice, there is left-right asymmetry that is highlighted in the arrow, the blue arrow on the right. So it turns out that our hippocampus on the left and right hemispheres, they actually do slightly different things. And so in most studies we look into, not only in animal, but also in human studies, it has been shown that there is left-right asymmetry. Some tasks would activate one side more than the other. And this seems to be consistent with what we are seeing here. We see left-right asymmetry in the wild type that we don't see in the APP knock-in. Right? So I'm going to skip this because I want to go to the computational part. That's part 2. All right. So that was what we've been doing for the animal imaging. I want to switch gears and talk about what we've been trying to do for understanding EI balance using human imaging data. And for someone who came from a computational background, this part is really dear to me. So I'm very excited to share this part with you, and hopefully I have enough time to cover as much as I can. So what do? We turn to statistical physics. I've always been fascinated by thermodynamics and statistical physics because, of course, from a physics perspective, these are the fundamental foundations. These are foundations of modern physics. And so if the human brain is some kind of dynamical system, then it should also follow very similar principles as a modern statistical physics. In particular, there's something called the principle of maximum entropy. And for people who may not be familiar with this idea of maximum entropy, it goes something like this. I believe I borrowed this sentence from Wikipedia again. The probability distribution, which best represents the current state of knowledge about the system, is the one with the largest amount of entropy. And of course, we know entropy is randomness. So in layperson's language, this basically means that nature likes having choices. The Ising model, I don't know if you guys are familiar with Ising model, but Ising model is a classic statistical physics model of ferromagnetism that can be derived from maximum entropy model or maximum entropy principle, and surprisingly has been used to study neuronal firing for over a decade. So I want to review 2 articles that really inspired this line of work. The first article is titled "Weak Pairwise Correlations Imply Strongly Correlated Network States in a Neural Population." And this paper came out in 2006 in Nature, so 16 years ago. And the second paper is "The Simplest Maximum Entropy Model for Collective Behavior in a Neural Network." And this came out in 2013. I want to, in particular, highlight this first article, 2006 in Nature, because the first time I saw this article, I was absolutely floored that you could use a classic physics model to understand neuronal firing. So I want to talk about what they did here. They took 40 retinal ganglion cells from salamander retina and recorded simultaneously. Then if you think about it, given a very small time window, then cell responses are essentially binary, whether the neuron has fired in that small time window or not fired. So you can code it as either +1 or -1. +1 is the neuron has fired in that small time window, and -1 would be the neuron did not fire. Then what they did is they use a maximum entropy Ising model, which basically includes all pairwise interactions. And they showed that this model would give an excellent approximation of the entire network correlation structure. I'm going to highlight some findings and the first mathematical equation of my talk today. For people who are not familiar with Ising model, it is essentially a way to model magnet. You start with the energy function, and that's the Hamiltonian. You have 2 summations. The SI and SJ are the states of a neuron in this case, and it takes a value of +1 or -1. The J sub ij represents the pairwise interaction we talked about earlier. The first term, H sub I, this is a bias term. So you can think of the H sub I as the influence for individual cell, and then the second term, J sub ij, models the pairwise interaction. And then once you have an energy of a specific spin configuration, the energy is then translated to the probability of observing that spin configuration. And this is the famous Boltzmann distribution. And that's highlighted at the bottom. So the probability of observing a specific spin configuration is proportional to the exponential function of the exponent being minus beta times the energy. Then you have a normalizing constant, 1 over z, and that is to make sure that it's a proper probability density function. The z is sometimes called the partition function, and in most cases are intractable, of course. The beta is inverse temperature, 1 over T, and that's a fundamental quantity in thermodynamics, of course. If you look at the equations more carefully, you will realize that the lower the energy is for a specific spin configuration, the higher the probability it is to observe this spin configuration. So, okay, so you can see the arrow on the bottom right. They fit this model to their data. So they fit the H sub i's and the J sub ij's. And then once they fit the model, they plot the fitted data against the observed, and that's the red line highlighted. So you can see by modeling J sub ij, that's the interaction term, you get a very good fit between the observed data and what's predicted by Ising model. On the other hand, if you only fit the H sub I, you don't fit the J sub ij, then you end up getting this blue line. Let me see if I can get here. See this line here? I hope it's clear on Zoom as well. And that's if you only fit H sub I, and that's assuming all the cells are independent because you're dropping the interaction term. And you can see if you do that, then the fit is not good at all. It's very different than observed data. So the take-home message is that surprisingly, using a very simple physics model by binarizing the signal, assuming that each observation has either spinning up or spinning down, and you only account for local pairwise interaction, you end up getting an excellent fit for what is actually observed in the living neuronal tissue. I found it amazing research. So I was so inspired. I think I first came across these papers maybe about 5 years ago, 6 years ago. And then we wanted to do the same thing for neuroimaging data. How many more minutes can I go?
About maybe 10 minutes.
Okay, good. All right. So we turn to connectomics. I'm assuming everyone here knows connectomics, so I'm going to really go fast here. Of course, we have the structural connectome and the functional connectome. The structural connectome comes from diffusion imaging. The functional connectome in this case is resting state with the Pearson correlation using the Bull signal, all that standard things. I think on a high level, 30,000-foot view, we can think of the structural connectome as like the highway system, and the functional connectome would be the traffic actually, going on the highways. All right, so the only thing different between the Ising model view of connectomics is really now we have to convert ROI signal into +1 or -1. And we simply say that if it's +1, that means that region of interest is active at that time, and -1 would be that region is not active at that time. Same setup as before. We have the Hamiltonian, and then that energy is going to connect to our probability of observing a specific spin configuration using the Boltzmann distribution. What's even more, we simplify the model by saying that since this is resting state, we are going to drop the first term. We're going to drop the H sub I to make our model easier. So we will be only estimating J sub ij's at this point. So this is the only term we are going to model. Now we talked about Ising model. We haven't talked about how we connect this to excitation-inhibition balance, because the reason we did this, really, we wanted to make inference on excitation-inhibition. Let's think more carefully. Imagine if J sub ij is positive. If J sub ij is positive and we want to drop the energy of a system, of a configuration, then that means the spin product Si times Sj has to be +1. It's by doing that, and J is positive, we drop the energy. So what this means is when J is positive, it is a ferromagnetic coupling, because the two spins would be +1, +1, or -1, -1. So it's a coactive relationship. If it's coactive, it's more likely to be excitatory. On the other hand, if J sub ij is negative, it is anti-ferromagnetic coupling. It's more likely to be inhibitory interaction. So using this machinery, we can now, by fitting a Ising model, we understand on a connectome level the amount of excitation inhibition balance. And how do we convert both signals to spin configurations? It's actually really simple. We do a global signal regression, and after that, we set the threshold to be 0. If it's above 0, we set the spin configuration for that ROI to be 1. If it's below 0, we set it to be -1. And we are definitely not the only one doing this. I know Danny Bassett, her group has been doing something very similar, and I believe they do exactly the same thing. So how do we compute the J sub ij's? Right now, we talked about the basic setup but we haven't talked about how we compute it, how we determine the pairwise interaction, J sub ij. And once it is determined, how do we sample the system with respect to the Boltzmann distribution? So this is the idea. We are going to take the structural connectome as the prior, and that's on the bottom left. Then we use this prior, we constrain our J matrix using this prior, and then we do a constrained maximum likelihood estimation. Once that is done, we draw samples using Monte Carlo. And this J matrix, since it is constrained using the structural connectome, but its dynamics exhibit that of resting state, that in a way you can think of this J matrix as the resting state structural connectome. It's like a hybrid connectome. And we like this, this term, resting state structural connectome. So when I say resting state structural connectome, I basically say the J matrix after we fit the model. So mathematically, this is what we do. Very standard maximum likelihood setup. You want to find the J matrix and the inverse temperature beta. These are the two parameters that need to be fitted. And J is, of course, a matrix, such that the probability of observing the overall spin configuration, the capital S of T, across all time points, the overall probability is maximized. So it's a standard MLE estimation. Then the constraint comes in as follows. We say that the magnitude of interaction between two brain regions should be proportional to the structural connectivity between them. And this is, of course, informed by the tractography. So here, coming back to the Hamiltonian, the energy function, we dropped the h sub I. We only have the interaction term. And now we further constrain the absolute value of j sub ij to be proportional to what is in the tractography, the edge weights coming from the tractography. So that becomes a penalty. We penalize that, deviation if the magnitude of J sub ij is substantially different than the structural connectivity. That gives you a penalty. And because the partition function is intractable, we have to convert it into a pseudo likelihood. Because if you do the original likelihood function, you try to differentiate, and you got into the problem that you can't differentiate the partition function because you simply cannot compute it. So we approximate it using a trick. It's called pseudo likelihood function. Then with that, we finally arrive at this gradient descent. The first term comes out of the partial derivative of the pseudo likelihood, and the second term comes from the constraint. And this is what we did. All right, so to summarize, we take our structural connectome that's on the top left, that serves as the prior. We fit our constrained pseudo maximum likelihood. Then we get the bottom J matrix on the bottom left, and that's the resting state structural connectome. And then now we treat that as an Ising model. We draw samples using Monte Carlo. You get simulated dynamics from this resting state structural connectome. And then you can see that the simulation is now pretty much the same as the observed. So it's the entire pipeline. And the way we compute excitation inhibition out of this is to simply do simple counting. We count how many positive edges there are in the J matrix, and we count how many negative edges there are in the J matrix, and that becomes our EI balance. Really simple counting procedure. This line of research is spearheaded by postdoc Igor Fortel. These are the papers that he has published recently in this line of research. He demonstrated that there is shifting EI balance in ApoE4 female carriers. And I'm going to briefly highlight these findings, and then I will conclude my talk. So the first data set he looked into, it's 76 healthy subjects, half of them ApoE4 carriers. And I think 60% of them are men. It's a relatively small data set. All these are healthy people. They may be carriers, but they are cognitively normal. Each of them also completed a water maze task, which is of course a memory, spatial memory task, really probing hippocampus. I'm gonna skip all these. So the findings from this data set is that indeed ApoE4 women or female carriers exhibit statistically significant hyperexcitation compared to non-carriers. And this global shift is less significant in men. Again, the main takeaway is that my belief is that the reason why women are at a higher risk for Alzheimer's disease may be related to hyperexcitability. That there's a differential hyperexcitability that you see in women that is not in men. So I'm going to skip all that. He's now replicating. Before that, so the degree of hyperexcitation in this data set is associated with this water maze task performance. Again, only in women. He's now replicating this finding in a much larger data set. It's called OASIS, has 1,041. And again, these are all healthy subjects. About 600 of them women, 400 men, and 200 of them are women carriers of E4, and 120 of them are male carriers. You see the same thing again. There's subtle but statistically significant hyperexcitation, in particular for the women carriers compared to the women non-carriers. So with that, I am pretty much summarizing part one of my talk before I come back tomorrow. Just briefly, I talk about how we infer excitation-inhibition balance using statistical physics, using mathematics, and Ising model, which is really a classic physics model. And I talk about how this is related to Alzheimer's disease pathology and how this may explain 6 differences in Alzheimer's disease. And with that, thank you so much for your attention. Oh, and this is my lab.
Thanks, Alex. Any questions?
Yes.
Can you test the model also perturbing in other ways? For instance, you can use brain stimulation, increase or decrease excitability, and again, also pharmacological interventions, you can really test whether the model is really sort of tracking these changes.
That's such a great idea. I know here at UCLA, you guys are doing a lot of brain stimulation research, right? Yeah, that's actually an interesting idea. We should talk about this later. I know we have Can we reach hippocampus with a standard coil? Is it too deep for a standard?
Well, you can do it using connectivity. Connectivity between parameters and areas that are more accessible to brain stimulation, and then you can actually reach the hippocampus level.
Yeah. So if the Kepler study is positive, then I can see a well-designed brain stimulation to really target or potentially inhibit hyperexcitability in hippocampus may also treat early MCI. That's a very, very interesting idea. Yes, thank you. Yeah.
Do you mind turning on your video on Zoom?
What is it?
Do you mind turning on your video on Zoom?
My video?
Yeah. So let me just go here. Okay. Yeah, the problem with Zoom is how do you get out?
Let me end and show.
Let's see.
Why don't we just do escape? Let's see if that's going to work.
Oh, wait a minute. You have a sticker on your camera, so never mind.
I see. So all this time I was not okay. All right. So it's always good to put a face to a name. So here I am. Sorry about my sticker that's blocking the camera. So
Any other questions? Yeah, sorry, you were saying.
Oh no, I'm just saying that it's always so good to be. This is my third in-person talk since COVID, and there's always so much more energy and human interactions. No pun intended. Human peer-wise human interactions, and it's just so intellectually stimulating. So it really has been a pleasure, and hopefully tomorrow I can continue talking about our smartphone research.
Not yet. Okay.
I have a question. So the Icing model is really interesting. It seems like a nice simplification. You know, a lot of features become binarized and things like that, and those tend to be robust. But computing these edge weights of J, it seems to be a pretty complex procedure. I'm just wondering if you looked at something like the sign of the correlation, something simpler, how does this more complex calculation relate to, adding up the positive correlative voxels and subtracting the negative correlative voxels?
Yeah, so that was actually the first thing we tried, because that was the most logical first thing to try, and it did not work. And another very strange thing, and it's really by design, if you think about it, since a lot of us, we do global signal regression with the both signals before we form the Pearsons. I know some people argue that we shouldn't be doing global signal regression, but at least if you do that, then you're essentially centering the correlations. So by definition, if you just just sum up all the values, you get roughly 0, right? So that's by design. So again, this global signal regression is also, I think the bottom line is there's so many steps involved in computational neuroimaging research, and tweaking one component can really have downstream effect that we don't know about. But yes, we tried that as the first step and didn't seem to work. And that's why we started thinking about fitting more complex models. If I may, I think one possibility why simply looking at the signs of the interaction, the Pearson correlation didn't work, I think it's also because the Pearson correlation is a fully connected graph. It's not really a graph per se, it's fully connected. So sometimes, even if there is no direct connection between two regions, you still get some kind of Pearson correlation. And that's why I believe that's why fitting something like Ising model forces you to think about pairwise when there is really pairwise connection according to structural connectivity. That gives you a way to filter out the sort of the secondary influence from a different node. This is my interpretation. So that's a great question.
So for the you used the MLE method to actually optimize your function, and then you do NCNC for sampling. But you could also use NCNC to actually optimize the objective function. So I don't know if you have any thoughts about, because I mentioned your cooling temperature beta, which you also estimate instead of setting like a scheme. So
Yes.
thoughts about that.
Yes, so that's a great observation. So I was simplifying the workflow a little bit. In reality, it's a lot of back and forth, right? We have to somehow guess the temperature first. And there is also a scale for the structural connectivity that we didn't talk about. So the inverse temperature is a scale itself. The scale for the structural connectivity is another scale. So we have to combine them. And then when we simulate the data, then we have to use a simulation to also readjust the scale. So there's a lot of back and forth. Yes.
Okay, so then let's thank Alex again.